Okay, so it's an honor to present once again for Covaris some of the work that we have done as part of the high throughput project that I'm doing with Jenny Van Eyck in Cedars-Sinai. And so today, thank you for participating. I know it's early morning. But I will be discussing the use of their Assay technology in the development of a fully automated, robust, high-throughput cell-based assay system that I've been working on for the past 2 years, essentially for protein extraction, and how I use that assay to then investigate a biological perturbation, and moving forward would be using it for other cell lines. Okay. Now, so in this presentation, I'll be talking to you guys a bit about the study rationale and some background context of our work, and essentially follow this up with our main bulk of the study, which is the development of the fully automated high-throughput system for the assays, and then discuss how we basically tested that system for stability by investigating a biological perturbation, and then follow that up with a short summary of our findings. And finally, highlight some of the future directions of the studies and the next directions of the project. Okay, so as part of our ongoing efforts in accelerating hard research in our group at Cedars-Sinai, we routinely use proteomic approaches to tackle the current challenges in precision medicine. And this primarily focuses, of course, on disease prevention, but can also aid in treatment and therapy approaches when you consider the variability in the genes of different individuals. Okay, so now, as I previously mentioned, one way of addressing this is through proteomics, where essentially a disease risk can be determined based on the quantification of an individual's proteome. So, to be able to accurately quantify these proteins, come up with putative mechanisms for discovery, and of course, generate relevant clinical data, there is therefore a need for an established high-throughput cell-based screening system. And this workflow can essentially enable the rapid identification of biomarkers, but perhaps more importantly, provide an individualized-based treatment regimen for each affected patient. Therefore allowing the therapeutic targets for early detection of cancers, improvement of drug delivery, and of course, real-time drug monitoring. Now, our previous work has basically involved the development of a high-throughput cell-based assay system via bottom-up proteomic approach. And from that, multiple protein extraction workflows were established for protein coverage. Now, in this presentation, I'll be talking about how we actually went one step further to make it really fully automated, which I will discuss in the latter slides, where essentially you can culture the cells in a 96-well plate and directly perturb the cells. And then, of course, from then onwards, you can actually test different drugs and different concentrations of drugs whilst the cells are being growing, and then, of course, do the whole sample prep and analyze it through LC-MS. So, in the next few slides, I'll be talking about how we actually transitioned our previously developed high-throughput cell-based assay onto this automated workstation. Now, since the hypothesis of our project revolves around cardiac perturbations, the assays involved in our project were cardiomyocytes. Now, cardiomyocytes are actually quite difficult to work with, and these primary cells, directed from tissues, you have the problem of fragility, and AC16 cells were therefore selected for the high-throughput cell-based assay. So bottom left, you actually have a schematic representation for the generation of the cell line, which is essentially a fusion between primary cells from adult human ventricular heart tissue and the SV40 transformed human fibroblasts. Now, the fact that it has fibroblasts, it makes it quite complicated in terms of the surface area that it occupies when you start to grow it, because it's quite big, and there's so much you can actually grow in a small well and a 96-well plate, which I will discuss in the next slide. But from the table here, you can actually see that it expresses most of the primary cardiomyocyte markers, and when you essentially compare it with a kidney HEC293T cells, which is the common cell line we use in our lab, it actually shows good expression of alpha and beta MHC class 1 and GATA A4 genetic markers. So, as I mentioned before, each cell grows differently and to varying cell densities, and therefore occupying a given surface area within the medium that it is being cultured. Now, as I mentioned, because cardiomyocytes are quite large compared to the other commonly utilized cell lines, we had to figure out a way to actually transition this. So, to initially understand the growth pattern of these cells, we first grew the AC16 cells in a standard 15 centimeter plate and assayed about 250,000 cells per well via the Covaris-Integrated system. And this basically allowed us to determine the upper limit of cells that could optimally be lysed by the AFA ultrasonication. And so once we established 100,000 cells per well to be the optimal cell density, we started to grow our cells in a 6-well plate. And then did a bunch of other optimizing experiments and managed to finally optimize that for downstream processes to yield the required amounts of peptides for bottom-up proteomic analysis. But that still wasn't enough, of course, because we wanted to move on to the 96-well plate for biological perturbation. So after several experiments later, we also managed to successfully optimize the required starting density, and basically succeeded to yield comparable amounts of previously established protein extraction workflow, which I will discuss in the next few slides. So, as I previously mentioned, a high throughput system would therefore allow the rapid processing of many samples with minimal volume requirement, enabling incorporation of more conditions per sample. And to essentially achieve the full lysis, we really needed to integrate our workflow. Of course, the Covaris is there, but we also needed a automated workstation, and we have the Beckman workstation there. And the idea was quite simple, really, to implement every step of our sample preparation after the cell culture phase onto a high throughput manner, where you could actually have 96 samples processed rapidly via the i7 workstation and transferred to the TPX Covaris plate onto the Covaris ultrasonicator. So to summarize this section, we wanted to achieve a greater throughput with reduced manual handling and increased data quality to then apply our high throughput system to biological studies. And of course, to achieve this, we went about and assayed 15,000 cells for 20 hours at 37 degrees in a 96-well plate and observed a 50% confluency, and that's really the minimum amount of confluency you would want, at least, for a good transfer. So when you determine the protein concentration, you actually notice that you do get about 0.3 microgram per microliter of protein that was recovered in a total amount of 6 microgram lysed per well. And this was a good starting point, which gave us enough data. But but this was still half of the amount of protein that we recuperated from growing the cells manually from a 15 centimeter plate. So we then went about to further optimize this. And several experiments later, we managed to establish a starting culture of 25,000 cells for 20 hours, yielding the approximate amount of protein from our manual approach. And that came, comparable to what we expected from the manual. And so this essentially concluded our integration efforts for harvesting the cells via the Beckman workstation with the ultrasonication and the Covaris system. Okay, so now here we actually have a bar chart that represents the quantification of the proteins. And so what's interesting is that you can actually see at least 2,000 protein IDs across the 8 biological replicates, with three of the replicates detecting up to 3,000 proteins. And this was fantastic because it was also the upper limit of detection from our previous experiments assayed manually. So this was a very good result for us. And on the Venn diagram here, you can actually see that you can observe about 80% of the proteins that is shared between the replicates from both of the approaches. But you do see that some of the replicates from the cells grown in the 96-well plate have some unique proteins. And this is another discussion that I had with someone, and this variability is not uncommon, as these cells were assayed directly from a 96 well plate culture, and the variance is really attributed to the biological variability as opposed to the technical stability of the high throughput system. So on the right, you can actually see the dynamic range of the proteins, where each is ranked against its average intensity. So despite the medium coverage, because we actually we did the LCMS on a Thermo system as opposed to the Bruker system that we also have, but that was occupied with a different project. So you would actually get a larger protein coverage, but it still proved about 7 orders of magnitude of different proteins, and that really demonstrated the diversity of the proteome that we can actually detect from our Covaris-Integrated platform. And that was really, really interesting to see, given that everything has been automated so far. Okay, now our established workflow is split into 4 key stages, and this essentially includes the cell culture, cell lysis, peptide isolation, and the liquid chromatography mass spectrometric analysis. So in cell culture, the desired cells are seeded to 75% confluency, and then you can actually treat it with a drug at a specific concentration per sample following the perturbation. And this can typically range from a week to 2 weeks, depending on how many drugs are being used to treat the cells. Then in cell lysis, so I actually have like a four chart at the bottom of that slide. You can actually see the 96 well culture plate that is being placed onto an automated workstation. So what the robot does, it removes the waste, and then the cells are washed and trypsinized for detachment. The resuspended cells are then transferred by the robot onto the Covaris-TPX plate, and then washed a couple more times with PBS before being resuspended in a detergent-based solution. And then the plate containing the resuspended cells are then sonicated by the LE220 Plus system for 2 hours and subjected to sequential reduction, alkylation, and digestion for bottom-up sample preparation. And at the end, of course, you have the digestion, so the resulting peptides are then dried in a speed vac and prepared for acquisition on the LCMS. That basically allowed us to understand that, okay, our system is now stabilized, and we can then go about to test this system to investigate a biological perturbation. And we actually tested hypoxia because it was also one of the projects that I was doing on the side. So to investigate hypoxia and reperfusion from ischemic heart injury, pre-cultures of AC16 cells were treated with cobalt chloride to induce the hypoxic state. And in this assay, the cells were essentially seeded for 3 days to 75% confluence, and of course, as I mentioned before, 25,000 cells per well were cultured in the 96 well plate. The cells were then perturbed according to the well-established ischemic reperfusion model, and then harvested onto our Beckman i7 workstation, which then allowed the cells to be lysed and transferred onto our Covaris TPX plate and sonicated for 2 hours, after which they were then, of course, reduced alkylation digested for another 2 hours on the i7 robot, and the resulting peptides were then loaded onto the LCMS. Okay, so following the LCMS data, so we use DIA approach. We performed this across 3 of the 8 biological replicates, and in this slide you will see the proteomic analysis of the cardiomyocyte and the results that we had. Top left, we can actually see the identified proteins for the 3 biological replicates, and you can see the normoxia condition in green, the yellow condition representing hypoxia, and reperfusion as depicted in orange. And you can see that there are actually about 1,700 proteins detected across the 3 groups, establishing a standard baseline for comparative analysis. And to the right of that, you actually see the normalized changes in the intensity of the data that is demonstrated before and after normalization to remove the random noise and variability across the samples. And then you have the correlogram, which basically shows the tight association of the data between the replicates. So the distribution of the proteins in each sample in the the smooth histogram really demonstrates tight the association is. Okay, so to the right of that, you actually see the detection of the protein IDs across 3 groups. And the left bar really shows the distribution of the proteins across the replicates. And the right side, you actually have the protein IDs that is present in 75% of the replicates. And I wanted to actually plot this because you can see the biological variability present in the reperfused sample, with the association between the replicates. Whereas when you actually account for the majority, with the protein IDs present in the majority of the replicates, you actually see that it's quite uniform. And then we further evaluated this on a Venn diagram, and we actually observed an absence of unique proteins in either of the groups, but we did get about 1,500 shared proteins that accounted for almost 75% of the detected reliable IDs. Okay. So to assess the relative dispersion of proteins in the data series around the mean values of each replicate in each condition, we did investigate the coefficient of variation, as with any other stability experiments. And when you look at the CVs, you actually see that approximately 40% of the detected proteins fell within the conserved range of less than 25%. With the reperfused state showing a slightly higher dispersion. On a principal component 2D graph, however, we actually observed that the replicates from each of the condition cluster independently and are present in different quadrants, with the replicates from the hypoxia condition actually showing the best association. And this was actually really good to see because you can actually see a very distinct difference between each of the 3 conditions, and that's exactly what we wanted to see in order to then do some biology on these experiments. And then I also have another plot of the principal component analysis. And again, this is basically based on the reliable protein IDs, as I mentioned, the IDs that is present in 75% of the replicates. So what's interesting here is that you actually see that one replicate from the reperfused and hypoxic sample actually associate with each other, which was really fascinating to see because, of course, both of those conditions are diseased. And you can see a clear, distinct separation between the normoxia control samples with the diseased sample there. Okay. So this slide shows the differential expression analysis of the proteins in a gene ontology annotation depicting the impacted enriched biological processes that is present on the left side of the slide, and the molecular functions on the right. So what's interesting to see again is many of the highlighted proteins here are involved in cellular proliferation and differentiation, but it was also interesting to see that some of these actually included some unfolded proteins, that is presumably led to dysregulation of mRNA metabolic processes, but it is actually detected with the with our Covaris-Integrated platform, which I found it quite interesting. Okay, so the diagram might not be as big for you guys to follow, but so we have a volcano plot on the left, and this really illustrates the vast number of differentially expressed protein that is lost in the hypoxic cells treated with the cobalt chloride compared to the healthy control cells, with approximately 15% of the upregulated proteins likely playing supportive roles following the oxygen depletion. Now the volcano plot in the middle actually represents the distribution of these proteins in the reperfused cells, where oxygen was resupplied after starvation. And the model really is that you basically induce hypoxia for 4 hours, and then you then basically treat them as a control for another 4 hours, and that basically represents reperfusion, because we really wanted to see what kind of effects you would actually get, if you actually reperfuse the cells. Okay, and so you can actually start to see some of shift of some of the downregulated proteins observed from the hypoxic condition into the healthy control cells here. And then when you combine, not combine, but when you compare the proteins from the reperfused to the normoxic group, you can actually see that almost the entire population of the downregulated proteins from the hypoxic condition are actually upregulated here. And this really further validates the capability of the high throughput system to actually observe this change. Okay. When you compare the downregulated proteins, we actually saw that they correlated with the previous gene ontology annotations, with most of the IDs displaying pivotal roles in RNA binding and lyase activity. Okay, so to better understand the proteomic changes in the ischemic cells, so what I did was I plotted the top 30 differentially expressed proteins against their respective fold change intensities. So on the left, you can actually see the upregulated IDs in red, and on the right, you can see the downregulated proteins in blue. And an interesting observation from the upregulated proteins in red is the detection of an insulin receptor 1 protein that had a log 2 fold change value of over 2. And this was the highest intensity amongst the other top ranked proteins, which was really interesting to see one protein being so different compared to the other upregulated proteins. But what's also interesting is in contrast to the upregulated ones, the downregulated IDs, most of them displayed a relatively similar log 2 fold change values, and this is really a direct consequence of the oxygen starvation in the hypoxic cells. I went even deeper, and so this is a heat map of the top 10 downregulated proteins in the heat map. And you can see that 7 of the downregulated IDs corresponded to ischemic indicators, and I actually saw all of these IDs being involved in hypoxic responses reported in several different literature and papers, which was really fascinating to see. Okay. So some of these examples include the enol coenzyme A protein, and so this protein is actually essential in fatty acid oxidation and is lost during hypoxia. But what's interesting is it's actually upregulated in reperfused cells. So we think that this enzyme is likely to be one of the first series of the proteins that gets expressed upon the reintroduction of oxygen in the reperfused cells, demonstrating the adaptability of the cardiomyocytes in ischemic heart injury. You also have the downregulation of the hemoglobin subunit protein that is involved in carbon dioxide transport, and several other cardiomyocytes that participate in mRNA modification, splicing, and GTPase activity. Okay. Now, as I mentioned previously, the insulin receptor 1 protein is here, strongly upregulated during hypoxia, and this basically activates certain cascades of metabolic pathways, which I will discuss in the latter slides. Okay, so we have a slide that represents the string analysis of our proteome. So sorry that the slides are quite small. I wanted to fit them in one slide. So on the left, you can see the top downregulated IDs, and I mean, you can see all the downregulated IDs, but the one in pink are the top downregulated ones. And so you can actually see the SYNCRIP gene encoding protein, which is a heterologous ribonucleoprotein appearing to interact with 6 of the other downregulated proteins in the network. Now, this protein is heavily expressed in the heart and functions in tandem with these 6 proteins in actually facilitating the mRNA modification and splicing, helping to induce cellular proliferation and differentiation, and this is lost in hypoxia. Now, on the middle network analysis, you see the upregulated proteins, and 11 of the IDs actually interact with each other in metabolic pathways, which I will discuss in the next slide. You also see IRS2, which is another insulin receptor substrate protein that actually interacts with IRS1, as I previously touched upon in the hypoxic cells. So on the far right, so what I did was I actually I combined the downregulated and upregulated proteins to see what kind of interaction these two have. And the interesting aspect here is that the impacted proteins in either direction have interacting partners working both synergistically and antagonistically in functionally specific biological processes. So for example, you have a TPT1, which is a downregulated protein that actually regulates cellular growth and proliferation. And this protein interacts with RPLP1, an upregulated ribosomal protein that becomes highly expressed in the oxygen depleted states. And this really made me understand that this assay could be used to really look deep into various different biological perturbation. Okay, so when we match the differentially expressed proteins on our in-house protein network interaction to map the biological processes, we actually observed multiple impacted molecular functions involved in DNA replication, mRNA modification, mitochondrial organization in particular, but also cellular responses to oxidative stress, which was directly pointed out. And only a handful of proteins were upregulated, most of which were involved in metabolic pathways. And this brings me onto this slide, where after looking into the downregulated proteins, we wanted to learn more about the small proportion of these differentially expressed proteins that were upregulated in the hypoxic cells. Now, the upregulated proteins are mapped onto another gene ontology enrichment analysis, where almost all of the impacted processes involved lipid and metabolic activity, which was quite surprising to me at least, all of which correlated perfectly with previously published studies and underlining causes of extreme oxygen starvation. And so I went to further explore some of these metabolic pathways. So here you can actually see the top differential expressed proteins mapped onto the specific lipid processes. And of course the result is demonstrated here. So what you can see is the majority of the IDs are all downregulated, as you would expect, and involved in lipase activity, phospholipid binding, and low-density lipoproteins, which, again, you would expect it with hypoxia. But the 4 upregulated proteins corresponded to high-density lipoproteins, and this could be a coping mechanism cardiomyocytes utilize to adapt to their oxygen-depleted state, reducing the accumulation of cholesterol and fatty acid buildup. And this could also explain why many of these upregulated proteins participate in lipid metabolism. So to summarize, I will now share some concluding remarks from our presented work today. So we have essentially scaled our protein extraction workflow to now a fully functional 96-well plate assay, and that has really allowed us to fully automate our Covaris-Integrated platform. Of course, optimize experiments to stabilize the high-throughput cell-based system to investigate biological perturbation. And finally, we showed that a high-throughput protein extraction workflow can be implemented to study the ischemic reperfusion cardiac injury. Which brings me to the future directions of this project. And we are currently exploring additional avenues for simplifying the protein extraction system. So the short-term goals include the development of a detergent-free workflow without purification using AFA technology and AFA-assisted trypsin digestion protein extraction workflow to enhance proteome coverage. And this is something that I've been working on for the past year. So hopefully, it will be out soon. And bypassing the, some of the sample prep workflows such as reduction and alkylation. And our long-term vision really is to integrate our high-throughput system to direct both label-free and stable isotope labeling workflows, as well as to adapt it to studying protein folding and post-translational modifications. Okay, so if you're interested in learning more about the high-throughput system, I have a post that I'll be presenting on Tuesday between 10 to 11, and my post number is P510. I have also attached a link and a QR code to the Covaris YouTube channel to access the previous breakfast workshop I presented last year at ASMS. So with that said, I'd like to thank the Covaris team, especially Samir and Eugenio for all their help, as well as Deb for generously allowing me to present today. And from our group, Max and Alion, Sample Prep and MS, and Alex for providing me the cardiac ischemic injury model, and other members of the Biomek lab for their support, and as well as you guys for listening through this presentation. So I'm currently in the process of submitting the manuscript, but I'm more than happy to take any questions that you may have. That's it.